Good day to everyone. My group will present about bioprinting of nerve. In this presentation, my group will explain the bioprinting based on biological self-assembly, structures of tubular bioprinting, and biofabrication of a nerve graft. Bioprinting is the use of 3D printing technology with materials that incorporate viable living cells to produce tissue for reconstructive surgery. In extrusion bioprinting technology, nerve conduits can be used directly as grafts to restore function after peripheral nerve injuries without the need of scaffold. The major components are bioink, biopaper, and bioprinter. The process of building the biological structure is divided into three well-separated steps. The first one is the preparation of bioink units, multicellular aggregates of specific shape whose cellular content is consistent with the composition of desired tissue or organ. In the second step, the discrete bioink units are delivered into the biopaper by the bioprinter. The biopaper represents a temporary support for the assembly of bioink units. It is not a part of the final product, thus it is not a scaffold. In the final step, the assembly of the discrete bioink units matures into the final continuous biological structure. This is the maturation step that is governed by biological self-organization.
Going to discuss about bioprinting based on biological self-assembly. So for the introduction, self-assembly is an autonomous organization of components from initial state to its final structure without any external intervention. So then the fabrication of the functional living structures requires the understanding of principles of cellular self-assembly for them to be employed. Then, for future biofabrication approaches, re-establishing the functional activities of damaged tissue function is important. Then, major components for biofabrication is bioinks filled with micropipette, uh, bioprinter, and hydrogel biopaper. Then our main component is bioink. Bioink is prepared from cell suspension. It can be homogeneous where it is prepared from one type of cell and also can be heterogeneous where there can be mixture of cells. Then it can be spherical and also cylindrical in shape. It also contains carrier material to stabilize it. Then it should be biocompatible. Then we are going to see the methods for preparations. Bioink. First method is egg holder aggregate maker. So as the first step, small wells will be created in an agarose mold using plastic templates. Then the cell suspension will be pipetted over the holes in agarose mold as soon as possible and will be incubated for 2 days. During this time, cell will settle down in the holes and will form spherical cell aggregates. Then second method is centrifugation. So cell suspension will be centrifuged and the resulting pellet is transferred into a capillary micropipette and incubated at 37 degrees Celsius. The cell-to-cell interactions will be restored and the cylindrical slurry becomes sturdy enough to be extruded into liquid. Then the slurry will be extruded from the micropipette into a non-adhesive mold where it is left to mature overnight to improve the block's cohesion. Then the bioink units are aspirated into the micropipette printer cartridge before deposition. Then we are going to see the bioink deposition and structure formation. So the micropipette printer cartridges are inserted into special purpose bioprinter equipped with single or multiple printing heads. In the case of multiple heads, the temporary support material, which is inert biocompatible hydrogel, is printed along with the cellular material. Then 3D structures are built layer by layer. Then the multicellular bioink units need to be stabilized until there is connections formed. Then to support the assembly, discrete agarose and bioink units coordinated and proceeds according to a template. Once the fusion is complete, the agarose is peeled off the cellular construct, which subsequently is transferred into a bioreactor for further maturation.
Next is bioprinting tubular structures. Before that, let's see how dope is formed in 3D bioprinted tissues. Firstly, neurostimulation is applied during in vitro maturation to allow the organ to improve or develop. Once the in vitro maturation is completed, it will be implanted. So this enables the vascular network to easily connect it to the host vasculature and also favors rapid integration by preventing hypoxia and cellular death. Finally, nerves will run along blood vessels in vivo. Here, the important note that we need to take account is that creating tubular networks that mimicking vasculature and neural networks has to be achieved before highly vascularized and innervated fully functional engineered tissues can even be contemplated. Thus, the first step toward the development of vascular or neural tissue is developing a tubular structure. Based on the research that has been carried out, the first method used was building a tubular structure vertically with spherical units. There are several steps involved in this method. So the first step is a sheet of biocompatible hydrogel is printed. This hydrogel sheet is made up of collagen, which support the deposition of bioink building blocks. Then, the alternate deposition of layers of hydrogel and building blocks is pursued until the desired height is reached. Finally, when the building blocks are fused, the hydrogel sheet will be removed and it will result in hollow tube as shown in this figure. There are some limitations of building a tubular structure with spherical binding units. The first limitation is using collagen as hydrogel. Since the success of printing is dependent on the control of the gelation state of the different collagen layers, the uneven gelation compromised the spatial accuracy. Not only that, collagen was also remodeled by the cells and partially integrated into the final structure. This causes difficulties in removing the hydrogel sheet. The second limitation is shape and size of the tubes, as it has the limitations in constructing the large size tubes and also complex patterns. The third limitation is due to the usage of spherical bioink. This is because the preparation of spheroids are time consuming and also filling the printer cartridge was difficult as the spheroids need to be aspirated one by one without a gap between them. In order to overcome those limitations, a second method was introduced in which the template for a tubular structure is built using agarose rods and multicellular building blocks. In this method, the agarose rods were used instead of hydrogel, and also the cylindrical building blocks were used instead of spheroid building blocks. In order to build the tubular structure, the agarose rods of similar diameter and the cylindrical building blocks will be deposited layer by layer to build the tube. Here are some of the examples of tubes that are built using this method. So these are the straight tubes of human smooth muscle cells of different diameter that were printed using 6 and also 13 multicellular units. Here, 1 and 7 agarose rods are placed to create the lumen of the smooth muscle cells. Then the multicellular units will fuse within 2 to 4 days. Lastly, the supporting agarose rods were removed to create structures like this. The second example is a vascular tube displaying a double-layer wall that is constructed to mimic the blood vessel structure. So in order to construct this structure, human smooth muscle cell and also fibroblasts were used as the building blocks. As we can see here, the two building blocks were fused but the two cells remain segregated. The third example is a multi-channel tube that is built by replacing some of the lumen-forming agarose rods with multicellular units. In this case, stem cells were used as the multicellular units to replace the agarose rods. Moving on, these are some of the advantages of using agarose rods instead of hydrogel sheet. So the first advantage of using agarose rod is it is a biocompatible inner gel that keeps its integrity during post-printing fusion and easy to remove. Thus, it serves as a temporary support without being part of the final product. The second advantage is the physical properties of agarose rods. It is a low viscosity liquid at 55 degrees Celsius and also easy to draw it into the printer cartridge. Therefore, it can be easily extruded as a smooth and also regular cylindrical unit. The third advantage is the preparation of the agarose rods. The temperature-driven gelation process allows forming the agarose rods of precise and also reproducible dimension directly in the printer without the need of adding chemicals.
Next, I'll be explaining about the biofabrication of a nerve graft. Firstly, let's look into the overview of tissue engineering of a nerve graft. The goal of a nerve repair surgery is to minimize loss function due to nerve injury, and a nerve guide is required to bridge the gap between the two ends of the injured side of the nerve to properly regrow axons and maximize the return of motor and sensory function. And currently, the gold standard for nerve repair requiring a conduit is an autograft. However, this type of repair implies the morbidity to the donor side. Therefore, efforts have been focused on developing a conduit that biomimics the properties of nerve tissue while minimizing their drawbacks. So, an ideal nerve graft should be permeable and biocompatible, provide a path for the growing axons, minimize the risk of immune response, and be able to accommodate neurotrophic factors. And currently, the majority of grafts in use are constructed from natural or synthetic polymers intended to mimic the extracellular matrix, such as the polyglycolic acid and biological material collagen. Aside from the raw materials to produce the graft, studies have been conducted in the effectiveness of seeding different cell types in the graft, such as Schwann cells, as it is the most common seeded cells known to improve the axon regeneration. And other than that, an alternative approach is to use a scaffold-free method which utilizes the self-organizing and self-assembly properties of cells and tissues to develop their own extracellular matrix. Next, let's look into the biofabrication of a nerve graft by bioprinting. So this diagram here shows the layer by layer depletion of cellular and agarose rods to form a 3-lumen tube. So firstly, since bioinks exclusively composed of Schwann cells are known to have weak cell-to-cell adhesion, the bone marrow stem cells were chosen as additional source of support cells together with the Schwann cells to form bioink units to form sturdy cellular cylinders. And as it can be seen in the diagram here, the bone marrow stem cell forms the most external layer of the tubular conduit, while the lumen of the tube is partially closed by the addition of 4 cellular units composed of 90% of bone marrow stem cells and 10% of Schwann cells and 3 agarose rods of 500 mm diameter for this particular nerve graft. And the agarose rods acts as a temporary spacer for the acellular channels and as a surrounding support structure. And then the tissue fusion happens. So the fusion of cellular cylinders leads to the formation of a tubular structure with 3 longitudinal parallel acellular channels. And then, after the maturation, the agarose rods are removed and the resulting multi-channel construct is ready for implantation to bridge a nerve gap. So as it can be seen in the picture here, so this is the final cross-section view of the 3-lumen tube. Moving on, let's look into the selection of the bioink and structure of the graft. So, the design of the bioprinted graft described earlier was guided by the following considerations. Firstly, the choice of the nerve injury model for implantation, and secondly, the creation of a multi-lumen pattern to increase the luminal surface area for axon regeneration, and then the cell type to support the axon regeneration. So, for a nerve graft, the geometrical parameters such as the wall thickness, the diameter, and the number of channels of the bioprinted nerve grafts can be easily controlled and modified to fit the injured nerve diameter and the length of injury. While for the cellular composition of the nerve graph will be defined by the two criteria, such as the ability of the cells to support the axon growth or the axon regeneration, and secondly would be the cell density of the supporting cells. As shown in the previous examples, the Schwann cells were mixed with the bone marrow stem cells, where the latter being able to produce robust cellular cylindrical units and also support axon regeneration. So, the failure of nerve guidance conduits to promote axon regeneration using nerve grafts can be due to some of the reasons, and some of the reasons are the lack of longitudinally oriented structural features, and it could be due to the low density of supporting cells such as the Schwann cells, and it could also be due to the immunological and inflammatory responses triggered by the scaffold itself.
Good day to doctor and my friends. I'm Kauslia here and I'll be talking about the post-printing maturation of the graft, which is a method that describes about the implantation of the graft with an animal model and also the functionality test which will be conducted on the graft after the implantation. Firstly, in order to create a three-channel nerve graft, cylindrical bioink particles, which are comprised of cells that assist neuron regeneration, and a cell inner temporary support structure are printed. Over time, the cellular cylinders are then merged to create a single and continuous tube with the interior channels. Next, the support structure will be removed once the post-printing structure had formed, and the construct is ready for implantation into the laboratory rats. The The left sciatic nerve of the rats are exposed, and the transection of the next nerve can cause a 1 centimeter defect. After that, the bioprinted nerve tube encased in a collagen tube is used to fill the gap. The native sciatic nerve's free ends are intubulated and sutured to the nerve tube. As in control rats, similar procedures are followed up until to create the gap in the nerve. However, it will be repaired with an autologous graft or an empty collagen nerve guide. Following implantation, the graft's regeneration capacities are assessed using several nerve repair evaluation methods. The most reliable approaches for assessing graft effectiveness are axon counts and electrophysiology. For an accurate axon count, some of the bioprinted grafts, including the proximal and distal stumps, are extracted 3 weeks following implantation to check on the movement of the axons in the proximal stump to the distal stump by manual counting. After 40 weeks, to estimate the overall number of restored axons, axon density across the graft cross-section can be determined as well. Bilskowski staining is employed as a standard metric of healing in the 3 weeks and 40 weeks time to see the axons. In the end, biofabricated graft shows higher density of axons near the lumina, where the cylindrical bioink unit containing supporting cells are located. However, the presence of agrosa within the channel prevented regrowth through the lumina. As for the electrophysiology method, electrophysiological studies are performed to evaluate motor and sensor recovery on rats with repaired sciatic nerves by different types of grafts, which are autologous graft, collagen tube, and biofabricated construct. This 40-week study compared the data taken for the compound muscle action potential and also the somatic pressure reflex. For the compound muscle action potential, it is a recording data of muscle voltage response to electrical stimulation of the repaired nerve. As for the somatic pressure reflex, the simulating electrode are positioned on the nerve distal to the repair site and baseline blood pressure, and after that the data will be recorded.
In conclusion, peripheral nerve injuries with frequent occurrence in traffic accidents and battlefield traumas often resulting in debilitating conditions. However, unlike in the central nerve system, in the peripheral nerve system regeneration is possible. The regeneration process could benefit from the application of tissue engineering. Bioprinting could greatly facilitate the application of 3D technology in the fabrication of biomimetic nerve conduits to repair damaged peripheral nerves. This technology can be improved with the effective development of bioengineered autologous self-based nerve guide by bioprinting with functionality comparable to an autologous nerve graft. This can revolutionize the current clinical practice of treatment of human peripheral nerve injuries. The advantages are reduction of valuable operative time and causes, morbidity of harvesting human nerve grafts, and the improvement in the patient's quality of life. Thank you.